Hi, I'm Amr Alexandari. I'm a PhD student in the computer science department at Stanford. Today, I'm presenting our work that demonstrates how neural networks trained to predict transcription factor in vivo binding profiles can learn the thermodynamics of protein-DNA interactions. Transcription factor binding is the most fundamental event of gene regulation and leads to activation or repression of target genes in response to stimuli or to maintain cellular homeostasis. This event underlies many different aspects of human biology, including cell differentiation and immune responses. Transcription factors can have 1,000-fold or greater preference for specific binding sequences or motifs relative to other sequences. This high specificity allows transcription factors to regulate the genome in a sequence-specific manner. Yet there is only partial overlap between experimentally determined sites and motifs, and there is still a need for new approaches to infer the sequence elements and associated transcription factor binding events responsible for the chromatin accessibility at each regulatory element across the genome. In vitro TF-DNA binding assays can measure the intrinsic DNA binding affinity of individual transcription factors. Mitomi, protein binding microarrays, BET-seq, and HT-CLEX are assays that allow estimation of relative binding energies for libraries of synthetic DNA sequences with varying degrees of precision and throughput. All these in vitro data have been used to learn high-resolution thermodynamic models of DNA binding affinity of individual transcription factors. Complementary in vivo transcription factor binding experiments can profile genome-wide transcription factor occupancy. In assays such as ChIP-seq or chromatin immunoprecipitation followed by sequencing and ChIP-exo can produce genome-wide tracks of read counts such as these, where peaks can describe potential transcription factor binding events. However, it has been challenging to relate these in vivo binding measures to thermodynamic affinities, estimated from in vitro experiments. How do we use in vivo binding profiles to reason about binding affinity quantitatively? Similarly, in vitro-derived models fail to predict in vivo TF-DNA binding. We observe strong peaks without a good motif and areas with motif matches that have no peaks. So does this poor predictive performance reflect the need to specify extrinsic cellular factors such as genomic context, local chromatin state, cooperativity between factors for correct in vivo localization? Or are our current in vitro-derived models not developed enough to accurately predict binding? In this project, we focus on genomic sequence context, which is suitable for in vitro, in vivo, and in-silico analysis. We will show that deep learning models trained to map genomic DNA to base resolution in vivo TF occupancy profiles from ChIP-exo and ChIP-seq experiments in yeast and humans are able to extract binding affinities de novo from in vivo occupancy and can predict the effects of subtle sequence variations in and around core binding sites on relative binding energy as measured by the in vitro BET-seq experiments. And they can do so with remarkable accuracy. We start by training deep learning models to map 546 base pair sequences from the yeast genome to base resolution PB-exo and ChIP-exo profiles of FAW4 and CBF1. Despite the small size of the training set, which was less than 4,000 peaks, the correlation of predicted and observed counts across peaks in held out chromosomes is extremely good. Here I am showing the model performance on the test and training chromosomes for PB-exo FAW4. Next, we use deep lift to interpret the models. Deep lift is an efficient approach to decompose the output prediction of the model for any input sequence in terms of contribution scores of individual nucleotides in that input sequence. We visualize a sample region from a test chromosome. We look at the predicted profiles for the forward and reverse strands overlaid on top of the observed profiles. They are very similar. We also look at the importance scores. This model is working as intended, as evident by the visible motif in the importance tracks. Now that we have the models working, how do we extract binding affinities? We use the trained models to predict binding strength for millions of synthetic sequences which contained a high affinity core E-box motif, CAC-GTG, with systematic sequence variation in the plus and minus 5 base pair flanking the motif. To generate a prediction, we randomly shuffle a uniform sample of 100 different backgrounds from test chromosomes. Then the model predicts before and after the flanks and core motifs are inserted at the center of the backgrounds. The predictions for the log total read counts in the inputs window are stored. The difference in predicted log counts between the background before insertion and the background with flanks and core motif after insertion is saved as the delta log counts for the sample. Finally, the delta log counts are averaged across the 100 random backgrounds to produce a prediction. We found that the delta log counts from the models had remarkable agreement with the corresponding relative binding affinities inferred from the microfluidic experiments. This figure shows the correlation for PBXO FO4 model, and this figure is ChIP-EXO CBF1. Then we can use a few samples and simple linear regression to transform the predicted delta log counts to relative binding affinities. We get similar scales on both axes and high Pearson correlation. These results indicate that neural network models are able to implicitly extract binding energies from the in vivo occupancy profiles and can distinguish subtle differences in flanking sequence preferences of two yeast TFs that bind very similar E-box motifs. Okay, what about human cells? Human transcription regulation is far more complicated than yeast. Well, our approach worked there. We trained similar models to map 1,346 base pair sequences from the human genome to base resolution in vivo ChIP-seq profiles of the glucocorticoid receptor in the A549 human cell line. Unlike the yeast TFs, the model showed moderate correlation between predicted and observed occupancy for peaks in held-out test chromosomes compared to replicate correlation, suggesting that the local sequence cannot fully predict in vivo occupancy of this TF in humans. Next, we used the trained neural network to predict occupancy for hundreds of synthetic sequences containing the glucocorticoid receptor consensus motif sequence with sequence variations at 1 or 2 random positions in and around the motif. Remarkably, we found that these predictions from ChIP-seq neural network model had very strong agreement with the corresponding relative binding affinity inferred from the in vitro microfluidic experiments. These results indicate that even for human TFs, the neural network models are able to extract binding energies from the in vivo data, despite the fact that these in vivo measurements contain extraneous signal not encoded in the local DNA sequence. We have shown that we can recapitulate earlier measurements. We can now go on to explore a fuller and wider range of effects and mechanisms of transcription factor binding that go beyond motif recognition. We do so through in-silico experiments. One debate we could shed light on with large-scale in-silico experiments is motivated by prior work by Affinities et al., showing that repetitive sequence may increase binding in vitro. In this simulation, we use the PBXO FO4 model and add an increasing number of CT repeats on either side of the CAC-GTG motif. As we increase the repeat length, we observe a generally favorable effect on binding. By aggregating the data from different simulations, we can observe various effects of repeat length on binding. The figure shows the favorable effect of CT repeats reaches a maximum around 15 base pairs of repeats, then saturates. On the other hand, GC repeats continue to have a strong effect up to 60 base pairs. The trends observed from the simulations strongly agree with smaller scale in vitro measures of binding affinity collected by my co-author Connor Horton. The measurements shown as points with confidence intervals demonstrate that repetitive sequences can modulate binding affinity in vitro in different ways. Our results indicate that deep learning models can extract thermodynamic affinities de novo from in vivo occupancy profiles of TFs. Our ability to extract thermodynamic quantities still applies in complex human systems, even when the model performance is only moderate. Using our models, we were able to decipher the influence of sequence context on affinity and in vivo occupancy. This biophysical interpretation of predictions of deep learning oracles of genomic TF occupancy opens a new avenue to study mechanisms of transcription factor binding via massive in silico perturbation experiments. I would like to acknowledge all the people who made this project possible, especially my co-author Connor Horton, who has been absolutely integral to this work. Thank you.
Oh, wow, that was a really interesting talk, Amr. Well, let's see whether there are questions here in the chat. Meanwhile, I will ask some questions. So I was curious whether you see differences between yeast and human, versus the contribution of the motif itself versus the flanks. So, and how much is, can you give me some idea quantitatively how much is coming from the flanks and how much is coming from the motif itself?
So in most of our experiments, all the synthetic sequences contain the core motif. So we couldn't really isolate the effect of the motif versus the flanks, but we can talk about the dynamic range of the different flanks. Yes. And it can have quite a large effect, actually, both in yeast and humans. Mm-hmm. I think in yeast, the flanks determined a lot more of the variation than in humans, but that could be due to the fact that in human cell lines, there are other factors that contribute to that variability.
So you mean in flanks in terms of things that are sort of like DNA shaped that are influenced transcription factor binding, or you also mean other motifs of neighboring transcription factors?
I am referring to proximal flanks that are almost touching the motifs, and I'm not talking about other motifs interacting.
I see. And so do you have a sense of, because you would expect that in the human genome, the interactions are more complex between different transcription factors. Do you see anything in that direction, or do you think it's similar in yeast?
Absolutely. I think in yeast, in CBF1 specifically, we can see the effect of having other CBF1 motifs nearby that can have a very large effect on binding. I think if there is also other work in the literature that shows that CBF1 likes to bind in pairs as long as they are within 100 or 200 base pairs apart, that can really derive an increased amount of binding. For GR and for FA4, I did not see quite the same effect. Mm-hmm. Yeah.
Okay. So I have two questions here from the chat. So Rahul Sitatan is asking, I found the part about repeat sequences enhancing TF binding interesting. Have you looked at longer repeats, including tandem repeats of the TF motif itself?
We did look at repeats up to 30 or 45 base pairs. Those are still can have quite a large effect on binding, and the model can still capture effects of repeats up to that length. Yeah, does that answer the question?
Okay. Yeah, and so Irina Kaplov is asking, could you use your approach to determine the effects of adjacent motifs of cofactors on TF binding?
That's the hope. I think it would be really cool if we're able to also start to capturing these effects of cooperativity and other TFs recruiting the TF. If we can capture that up to the same standard of in vitro experiments, then that would be really cool.
Mm-hmm. Okay, so here's another question, another interesting question. Anushka Biswas, I think who spoke yesterday. How do the binding affinities differ in case of direct and indirect binding transcription factors?
I see. I don't, that's a really interesting question. I don't know how people measure indirect binding affinities in vitro. So perhaps I can think about that a bit and then maybe answer that question offline.
I think what is meant by that is sort of an indirect fuzzy binding footprint. So when you have a transcription factor binding to a motif that it normally doesn't bind, but you still see ChIP-exo reads.
Oh, I see. I actually, that is something that Tie7 does. The Tie7 peaks are usually only present. It also binds a very similar motif to CBF1. It has the same motif in vitro. Yet in vivo, the binding is very different, and that's largely due to other TFs recruiting and having that kind of indirect binding between the two TFs. Mm-hmm. That is actually a TF that we're looking at next, specifically for that question.
Very cool. All right, so we're going to move on to the last talk of this session.